and go into babies and ask, are these proteins being digested? Now, this is where it's so obviously important scientifically to design an experiment to show when you're right, but more importantly, when you're wrong. Because I was absolutely convinced that premature babies could not digest protein. It was obvious. And I'd be famous. And the graduate student tested, and I was completely and utterly wrong. This is the peptides being released from the proteins of milk. The pink is the peptides going into a baby, and the blue is what's coming out. And the proteins are being digested beautifully. Wrong again. Then we went further. This just says that the proteins are being digested, but we have no idea into what. So what we did was began to take this apart. And the chemists, you'll love this. We start doing tandem mass spectrometry on every single ion coming out of those babies. Now, remember, when you do that, the daughter ion has such accuracy in the mass spectrum that we can literally sequence it. We have the entire genome, remember, it took us 10 years to do that. We know all the proteins are sequenced. We can take these peptide sequences and map them, where they're coming from. Now, I say that so cavalierly, but it had to be done one spectrum at a time, one after another, one peptide, to build a massive library. Thank you for graduate students. But now we have that. We have the full library. We have the library of all the peptides, and that means we can start interrogating all of these peptides. Where are they coming from? What's releasing them? So it turns out the digestion of milk proteins is very specific. Some proteins are not touched. Some are cleaved very specifically. And we know that from the peptide fragments, but we also want to know what are the enzymes that are doing it. Because remember, a protein is cleaved by a protease in very specific amino acid residues. And when you know what the residues are, which we do, you can predict what are the enzymes that are doing it. So Nora Caldy, a computational biologist, predicted absolutely conclusively these are the 5 enzymes that are breaking up milk proteins in the baby. Except babies don't have any of these enzymes. Where are these enzymes coming from? Juan Medrano sequenced the message levels in mammary epithelial cells. They're coming from the mother. Of course, milk is self-digesting. Again, a genius idea. So mothers are making sure that the baby gets all of the value of the proteins by enhancing the digestion, which is a wonderful idea. And now we understand something that's been puzzling us about the neonatal intensive care unit. So if a mother has a premature baby, and she can lactate and provide milk to her baby, then that baby gets her milk. But if a premature baby's mother cannot produce milk, which is not atypical, she's premature too, then that baby has to get banked donor milk. And it's been noticed for years that mothers who can give milk to their baby, those babies do better than the babies who are getting banked milk. But it's just milk. But what has emerged from that is this idea that premature milk is better for premature babies. That is evolved to be for premature babies. But unfortunately, that's nonsense. There didn't used to be premature babies. The mammary gland is in fact immature, and it's not producing ripe milk, but it's not actually of benefit. What else could be the explanation? Well, if the mother is making milk for her baby, they give it to him or her raw. But if you're getting donor milk, it has to be processed, pasteurized, to get rid of pathogens. It's heated. And what's sensitive to the heat? The enzymes. So the babies can't digest, in essence, the proteins. So now we know. So we're working to perfect ways to process human milk to get the enzymes to premature babies. But there's a larger issue. What this proves is digestion isn't easy. Milk proteins are very digestible, but still mothers provide enzymes to help the babies. So if you don't digest a protein, it doesn't vanish. It keeps going down the intestine. And when it gets to the intestine, it then becomes food for bacteria. And it's not just a food for bacteria, it's a selective food. It's a selective food for certain bacteria, unfortunately, proteobacteria gram-negative enteropathogens. And what that does is produce endotoxin, and you feel bad. What is the #1 most undigestible protein in the human diet? Gluten. Now we know about gluten intolerance. There's some people who have celiac disease, but there's a lot of people who can't digest gluten. And it's giving rise to these problems. What's the solution? Ask Yamada at Enzymes. So we're about to launch a company that's developing enzymes for every one of the food proteins that's difficult to digest. Again, learning what we can from milk. So what we've realized from all of this, that there are some key opportunities to change the way we eat. Guiding an intestinal microbiological community is not easy. You have to know who's there. What are the bacteria in there? What are they eating? If you don't eat in that community, you die. And what are they making? That's a complicated series of questions, but we can now answer them. In the case of adults, if we're going to replicate what we found for babies, it's going to be a huge challenge. Remember what we achieved in order to understand the microbial community in babies. Carlito Labriglia built a library of every single sugar, every single linkage, and all the oligosaccharides in human milk. David Mills built a library of all of the bacteria available, their genomes, and the genes associated with breaking down and consuming all of those free sugars, and mapped all of that to the metabolism of those bacteria to know what the metabolites were that were going into the bacteria and to the baby. We did all of that for babies, and it took us 15 years. If you're going to take the same principle to adults, you can't say eat fiber. That's not nearly accurate enough. You're going to have to measure every single sugar and every single linkage in every polysaccharide in every food you eat. And you're going to have to map all those sugars and those linkages to the bacteria that you're going to need if you're going to eat those complex polysaccharides and nourish the right bacteria. Well, how are we doing? We're almost done. Carlito has developed methods that we can now measure polysaccharides sugar by sugar, linkage by linkage. He's done already 200 foods. David Mills has built a library of the microbiological candidates to be able to digest all of those oligosaccharides. He's mapped them in terms of their community structure and for the metabolism of those sugars all the way through to the metabolites. So we can imagine the day when we can nourish people with polysaccharides, food materials, plants, and the bacteria they need to do that. We will not eat all the same. Some people will want this kind of food, another kind of food. We will match the bacteria to them. Personal, and you get the same kind of benefits that babies get. So it's an attractive future. It means more microbiologists and more microbiology in the food supply. It will have benefits. 40% of the world's food today is lost to waste. If we can improve the stability of food, then we can dramatically increase the available food supply. And the model is cheese and wine. Most foods, as they sit around, they deteriorate, they get lower in value. Cheese and wine. When they sit there, they get better and more valuable because of the microorganisms. So we are looking at a future where there's, in essence, microbiology as the next generation of bioprocessing technologies. And this will dramatically change. Safer, more nourishing, more stable, more delicious. The semiconductor of the 21st century is the bacteria. So everybody contemplating a career change, this is going to be your future. And we're building, in essence, an industrial park at UC Davis, and it's Startup Park to build the companies that will realize that future. Agriculture's role, now we've got to feed them. All these bacteria that we, in essence, are living with, we have to build the next generation of selective glycans to make sure we get the bacteria growing that we really want. So, what it means is a future that's both more specific, precise, and personal. And personal means we have to understand the diversity of people. So phenotype is a term for all of the various attributes of an individual. Their metabolism, their anatomy, physiology, cognition, and of course it's the result of genotype, which you got from your parents. And environment, all of those, in essence, diet, lifestyle, environmental exposures that you're currently exposed to. But for the phenotype of humans, it's more than that. Throughout our lives, our environment interacts with our genome, and we learn from it. And so our phenotype changes. So if you're an athlete, you exercise to change your phenotype. If you have a very poor diet earlier in your life, you will be at greater risk of certain diseases for the rest of your life. And that's because of this interaction between genotype and environment your entire life. So, perfect phenotypes, these are my daughters. They're partly the result of genetics, of course. They'd like to be tall, and they're not. And they know exactly why. If you want to know your flaws, have a daughter. She'll tell you each and every one of them. So they're partly genetics, but also their environment, what they choose to do, how they choose to live. And so we tend to think of personalization as being dominated by genetics. Well, it's not. We have tremendous elasticity in our phenotype due to diet. I will remind you, this is a daughter at 1 and 21, takes 10 million calories to make a perfect daughter. She's the exact same genetics at 1 and 21. Clearly a different phenotype. So the precision that we're going to have to deliver diets is not just genetic. It's genetic plus all the various phenotypic aspects. And we're going to have to alter our environment and monitor our environment, because that's how we interact with the world, especially microbiologically. Right? Today, our acquisition of bacteria is completely random and frankly not particularly well suited. So we have to know what the environment is. So it turns out, daughter was deciding between going to Boston for 3 years, 5 hours away by plane, or San Francisco, an hour by car. So her mother brought home a high-affinity daughter binding device. An 8-week-old golden retriever puppy. Daughter spent 3 years in San Francisco. We now have Bailey. But if you have a dog, it extends your lifespan more than Lipitor. And if you look at families and their microbiota, if they have a dog, the microbiota of the family is much more similar than families who don't have a dog. So the most influential contributor to the microbiological community in you is something we don't even think about today. The dog. All right, so conclusions. Thank you very much. I'm delighted by this. We're forging ahead.